vermicellar. They have large gougy bodies. Trichomonas vaginalis is one of them. Right here they have, that's Trichonympho. Here we go, Trichomonas. Right here. Trichomonas vaginalis, he has a few, they have a few flagella, and then they have an undulating membrane, so on and so forth. Not much that you have to worry about. But Trichonympho, the one that is found in termite gut, is that right? You all know what I'm talking about? Mutualism, parasitism, and then Spirotrichonympha. This is also found in termite, but we do not have this in the lab. But we have pretty much this one in the lab. Most the slides we have in the lab is that, and then of course we have this one in the lab. So the other one, Dianthamiba, we do not have slide of it, but it's another organism in this phylum. And Trichonympha, of course, Trichonympha vaginalis. I gave you the name of the species. You do know the name of, I'm sorry, Trichonympha, I'm not straight yet. And I did not give you the name of the species, it's just the genotype genes. Here they are, a few species in flagella, Trypanosoma. In old days, old days when I took zoology, these two were placed in the same phylum. Now, Protozoa phylum, when I took zoology, there were 4 phyla, but nowadays, God knows, I don't know, we're studying what, 10 of them, 5 of them, 12 of them. So that's the, this is Trichomonas vaginalis, and this is Trypanosoma, which they are not in the same phylum. Okay, the major taxa, the other one, phylum Dinoflagellata. It's other species of Dinoflagellata are harmful and toxic to many organisms. Coccidea, which is cause the red tide, has both amoeboid and flagellated forms. Among 20 body types, generally feed on bacteria and algae, where they can temporarily use energy stores in chloroplast. Release short-lived toxins. That's what we human, when we have you heard of red tide, I will show you a picture. Red tide, when we eat the crab or we eat crayfish, here we go. When we eat crab, crayfish, we are actually consuming the toxins that these organisms create. And then, of course, in some cases, people die. So what it is, it stuns and kills the fish. Of course, it's hazardous to fish as well, create lesions that affects fish along the Atlantic coast of the United States. Here is a red tide. That's what I'm talking about. And then what happens, you know, of course, the fisheries industry, they do not send the crayfish or crab to the market because of this. And when does it go away? When can we start eating the crayfish or the crab? Next season, next year. This year is gone. You cannot have the fish or crabfish or crayfish from this shores this year. What did it say? Makes a striking appearance on the coastline. Okay, here are a few other species that we have in the lab which belongs to this phylum, phylum Dinoflagellata, which is your serratio. You do have that slide. We have slide of serratio in the lab. Does not look, well, it kind of look like this, but this is a nice drawing of it. And the other two organisms, we do not have it in the lab. We have this one, serratio. Okay, finally, the last phylum is Phylum Apicomplexa. Look like a tank. Oh, you guys already know what is this in here? It's a tank. Yeah. They're all members of this phylum are parasitic. And then they have a structure called apical complex. So remember in somewhere at the beginning of Protista, I said that some species, some organisms, they have a special structure. So they have a special structure. This is, for example, the nucleus of the organism. They have another special structure right here, which they call that apical complex. It is collective of other structures inside of it. That's what they call it, apical complex. It's an organelle. It's an by definition, organelle in biology 1 is a membrane. Organelle is a membrane-bounded structure which has a function, metabolic function. That's the definition of organelle from biology 1. So they have apical complex, and there are two. I gave you two. There are more. I gave you two structures, rope trees and micronemes, in the apical complex structure, organelle, if you would. So in the apical complex structure, I gave you two names, rope trees and micronemes. Both, to keep things simple in this class, both of them, they are used to gain access into the host cell. So if you have a host cell here, okay, right here, you have a host cell, this is the nucleus of the host cell. They use those two structures, the microneme and rope trees, to dissolve the cell membrane of the host cell, and eventually you would have this one, we draw the exact same thing, you would have it inside of the host cell. So this organism, this apicomplexa, is inside of, this is a host cell, whatever cell it is, which we are going to study a few. So, and pseudopods, in some intracellular stages, it means these organisms, the way they move by the term gliding. They use the term gliding. So underneath of the cell membrane, you have these little microtubules, and in biology 1, you studied what microtubules are. Underneath, there are little microtubules. And what happens, it seems like it, based on what scientists are saying, the cell membrane, it glides over these protein. This is protein, you remember that from biology 1, which are microtubules, and this is phospholipid. Okay, so these phospholipids are gliding, if you would, over these tubules, those protein molecules which are called microtubules. And that's why I have the picture of the tank. The picture of the tank I have is an analogy. So this would be cell membrane, and these wheels inside, those wheels are your microtubules, the protein molecules. And that's how these organisms move, by gliding. Before we talked about pseudopods, cilia, and cilia 9+2, so on and so forth. There is one more mode of locomotion left, and that is gliding. So they call it gliding. So just think about it. Flagella, in some gamete stages, you will see in some species asexual and sexual reproduction. Yes, it's possible, both of them. And then some requires an intermediate host. And what that means, so far, you study a direct life cycle. What that means, direct life cycle, it means that giardia is transmitted from somebody's feces to a body of water or some fomites, right? We talked about this, and you put it in your mouth and then you become infected. That's how it is. Not necessarily somebody grabbed somebody's, but in animal kingdom, you know, they're exposed to feces of each other a lot. Us human is a little bit different. That's why we use the term fomite. I talked about that a little bit before. What happens intermediate host, it means a stage, you will see it here in a minute, the asexual reproduction cycle of this parasite takes place in a different organism than the sexual reproduction. For example, let's talk about malaria. I'll give you a prelude to malaria. What happens in case of malaria, which you all have heard of malaria, you know what malaria is. What happens, the sexual reproduction of malaria takes place in mosquito. And human is intermediate host. So malaria, a final host, or another name for it is definitive host. Final host or definitive host is mosquito. And human is intermediate host. Okay, I hope I'm making some sense. But that is in some species. Some species require intermediate host. You also we talked about trypanosoma. In trypanosoma, you do have a fly, city fly, we talked about that, or Chagas disease, sand fly, and so on and so forth. So the life cycle in those is not direct life cycle. They do have a vector. They call it a biological vector within. Some develop oocysts or spores, which we will talk about that. Here they are, the general characteristic. This is a cyst or oocyst stage. So oocyst wall, and then sporocyst residual right here, or oocyst residuals, sporozoites. So we'll talk about, I did talk about this a little bit before when I was talking about sporogeny and schizogeny. I talked about this. So inside of the oocyst, these nuclei multiply, and then you have, these are called sporozoites. And then this is the trophozoite stage, the feeding stage of the organisms. Look, all of the Golgi apparatus, nucleus, endoplasmic reticulum, mitochondria, and it has everything. And then, of course, in this area, they have the apical complex. And all of those apical complex, I'm hoping you would know rope tree and micronemes. These are possibly the largest, especially the rope trees is the largest structure in the apical complex area. Okay, the first class, again, I'd like to break it down for you guys. You don't have to know what this one, the name of the class, but the class is the most important class because human diseases are like malaria belongs to this class. A few other diseases that I will talk about, but they are all in this class. Okay, so that, why is it important? A quiz question, because a lot of human diseases are, animal and human diseases are in this class. Pyroplasma in cattle, it causes Texas water fever caused by Babesia bigemina. Babesia bigemina, what it caused the cattle to urinate blood. You have blood in their urine, and that's what they call it. Texas, first time it was seen in Texas, but it can be found anywhere. Texas red water fever. And then human also becoming infected with Babesia bigemina or other similar species, and it is what it is. It is the signs and symptoms are similar to malaria. So a lot of people, when you submit your blood they check you and they say, oh, you have malaria. Well, you don't have malaria, you have Babesia. So always ask for second opinion, second lab, a different lab. Go and submit your blood, and they might misdiagnose you. So they think it was Babesia, but it was not Babesia. Next organism is Cryptosporidium. Cryptosporidium it should come up. I have a slide. It caused diarrhea, severe diarrhea, and there were a few outbreaks in the United States, in Milwaukee, and in Washington, D.C. area. There are a few thousand people became infected, and a few people died as a result of it. But it is more severe than Giardia. Giardia, you don't get much attention, you drink enough fluid, you live through it. This one, some people, they die of it. And they have strong immune system. In case of Giardia, if somebody does not have strong immune system, HIV patients or AIDS, I should say AIDS, people with AIDS, Giardia can kill them. But people with strong immune system, Giardia does not cause much harm. Okay, well, it causes diarrhea, but you get over it. This guy, it is possible that the person can die. Okay, I hope I'm making. Coccidea, intercellular, they do access, that's what I mean. They get inside of the host cell. That's what I mean by intercellular. They get inside of the host cell and they cause the damage. Eimeria isospora cause coccidiosis in farm animals. I don't know, you talk to any farmers and they say, oh, this year I got coccidiosis. They're talking about these guys, Eimeria and isospora. Isospora in AIDS patients is, it can cause death. You and I probably will not even know what we have. Ameria tenali, I've heard horrible stories in people who have chicken farm or they have turkey farm. They get up, I've heard it, I have not seen it, but they get up in the morning and every chick, every animal in the farm is dead. Okay, of course I'm thinking small scale farms, the huge farms, it could happen. But that, what happens, we'll go over the life cycle here in a minute, what happens when a few animals are infected, they release diarrhea, and chickens, they eat each other's poop. So they come and pick on other animals' poop, and they're picking grains from ground, and they become infected as well, and they all die by morning. But that's what happened in the United States. They do, the chicken farms, they give them a lot of antibiotic that hopefully they do not come across as a prophylactic. And that's why other countries, some other countries, they do not buy United States chicken because they're full of antibiotic. And of course, it has effect on us, on human. So it's Eimeria. Eimeria life cycle is, as I said, is direct life cycle. Let me pull up the screen and talk about it a little bit. Okay, so what happens here? So you have inside of a chick gut. This is the gut, chick gut. What happened? You have Eimeria right here, and I will talk about how did they get it. So they keep multiplying, and by a process called schizogeny. Remember that? So you have one cell, multiply, and then cell membrane is formed after that, right here. And then eventually they put out these, and then, well, they multiply, and then you have the microgametes. I'm forgetting the steps. And macrogametes. They come together after schizogeny. You guys follow what I'm talking about? I'm not very organized on my drawing. Sorry about that. I don't want to erase it and write it again. And I will tell you how they get in the end in a minute. Hang on. It's a cycle. Wherever I start, it's still a cycle. So right here, they come together and they form an oocyst right here. So oocysts come out in the feces of the chicks. I want to make you some sense. So in the feces, you have oocyst, and oocyst in the environment undergo sporogeny. We talked about that. So one nucleus divides two. You have two. In case of Eimeria, I'm not sure. I think the maximum is 4. In case of Toxoplasma gondii, it's 8. We'll talk about that. And then these 4 will be ingested by another chick, a healthy chick. This one probably dies because they have severe diarrhea. Then another chick will ingest this 4. In case of Cryptosporidium is 4. Okay. Am I making some sense when I say 4 or 8? Okay. So in case of Eimeria, let's say it's 4. As I said, I'm not sure. And then when they go inside of, they pick it up from ground and they eat it. When it goes inside of the intestine of chick, each one of these 4 break apart from oocyst. And then you have 4 of them here, which they multiply by schizogeny. And they destroy epithelial cells of the intestine of the chick. So next chick, and it's very bad. Yes?
Is there any way to cure that in the chickens? Antibiotics?
Farmers give it to them before they get this.
Oh, okay.
Yes. Yes.
So where do the micro and macro gametes fit in the cycle?
Okay, good. So you have these, and they, can I enlarge this? Here is one of the sporo, they call it sporozoite. You have 4 of them, remember? There are 4 of them inside. So you have 4 sporozoites, and they get into epithelial cells, which you studied at the beginning of the columnar epithelial tissue. I draw the nucleus on the side. The nucleus is right here. So when they gain access here, right, then what happens, they multiply the nuclei, right? And then after that, they form the cell membrane around it. That's what they call it schizogeny. It's not mitosis, really. And then some of these guys become they destroy that cell, they get out of that cell, they go to next cell. Am I making some sense? So they go, then some of them become microgamete, and some of these become macrogamete. They come together in the intestine of, you will learn here in a minute, in the intestine of the chick. They come together and form oocyst. So, as I said, this is a direct life cycle. It doesn't need a mosquito. It doesn't need snail. It doesn't need human. Just from one chick to another chick to another chick. That's how it is. Okay? Shall I go? Should I go? Or should I stay? Yes, no, maybe perhaps.
Coin flip?
The what?
Flip a coin.
Flip a coin. Reproduction of Eimeria, I talked to, here we go. Schizogeny, I talked about it. Gametes, fertilization, oocyst, easy. And then in the intestine, in the feces, and then they get out of intestine, sporogeny in the environment, in oocyst, 8 sporozoites, yes, okay, and then outside of the host. It's outside of the host. And then what am I saying outside of the host? This is happening outside of the host. This is happening inside of the host. I talked about it, I wrote it on the book. Okay, so new host ingest sporulated oocyst, and voila. Toxoplasma gondii, I have a separate presentation for it. I don't know, maybe I get to it today, maybe not, we'll see. In AIDS patients, based on statistics, based on studies they've done, half of us in this room have this parasite. And we live with it until the day we die. It does not harm us. But however, if sometime or other, Aisha, in your life come across HIV, or you come across a disease that wipes your immune system, these organisms inside of our body go wacko. They multiply and multiply and multiply, and eventually they die. Many of you know Arthur Ashe. He was a great tennis player back in the 1960s. He was African-American tennis player, famous. He won a few championships. And it's rare in tennis world. It's rare. He became infected with Toxoplasma gondii early on in his life. Nobody knows when. You have no clue when you became infected with Toxoplasma gondii. Okay, and then later on in his life, he needed blood transfusion. He got blood transfusion, he got HIV, and then he died of Toxoplasma. Look it up, Arthur Ashe. Definitive host is cat. What is the definition of definitive host? I talked about it. I said something about it today. It's
Where
what?
they reproduce.
Sexual reproduction. Where the sexual reproduction occurs only in cat, all sort of cat, domestic cat, wild cat, the feline family, all of feline family. That's where sexual reproduction, you remember sexual reproduction, microgamete, macrogametes, right there in the chick intestine. I talked about in case of Eimeria. In case of Toxoplasma gondii, it is in the cat. Okay, possible intermediate hosts are us, pigs, birds, sheep, goat,
Most every.
most everything. Most all other animals. Plasmodium, okay, I will come back to Toxoplasma gondii, guys, a little bit later on in more detail, because that's what I did in Tennessee. I worked with Toxoplasma gondii, so I thought I'd give you guys a little more detail of what you need to know. Okay, Plasmodium, malaria transmitted by mosquito, sporozoites injected to human by insects, sporozoites in liver, initiates schizogeny, I already said that, I don't have to talk about it. So first place they go. First place, Plasmodium goes. Anytime I say Plasmodium, I'm talking about malaria. You all know that. So the first place they go is to your liver. Okay, and then they initiate, their reproduction, asexual reproduction, schizogeny, asexual reproduction, the nucleus divide. Then cytokinesis occur. You remember that? I talked about that. Okay, so in liver, and then Plasmodium falciparum. There are four Plasmodium that you should know, but the most devastating one is Plasmodium falciparum. We have Plasmodium malariae, Plasmodium vivax, we have Plasmodium and Plasmodium falciparum. Plasmodium falciparum is the most devastating one. It's the one that kills human. And the slides we have in the lab, most of them are Plasmodium falciparum. So what happens? They penetrate red blood cells after one generation in liver. So they go into RBC, and then RBC goes into liver. They get out of RBC and they penetrate. It's trophozoites feeding. They eat hemoglobin. HBG is my abbreviation for hemoglobin, and RBC is red blood cells. Sexual reproduction, microgametes and macrogamete happens in mosquito. Okay, sexual reproduction occurs in mosquito, ingest gametes, and then fertilization, oocyst. And this is what I'm hoping you will know. Let's go quick. Can you guys see what is up there? Huh? You all can see that. Is that in the camera? Can we see that? So as I said, this is a cycle, guys. It doesn't matter where I start. So let's start with, right here is the Plasmodium, the Anopheles is the name of the mosquito. Anopheles is the name of the mosquito. I guess I'm not holding you responsible for this exam, but when we get to exam number 5, then you should identify it on the microscope and you should know it transmits malaria. For this exam, just mosquito. I'm happy with the term mosquito. Mosquito infects human by injecting saliva right here. So right here, the injected sporozoites, they go into blood and eventually in the liver. Right here, schizogeny occurring in the liver, human liver. And after cytokinesis proceeds, what happens, they destroy liver cells and they get out, they go into red blood cells and they do the same thing, the schizogeny in the red blood cells, and they keep multiplying and multiplying and multiplying. And you know the signs and symptoms of malaria. A person has fever, they have chills, they have headache. That is pretty much when the red blood cells all of a sudden are destroyed. Not all of them, but when the red blood cells are destroyed, then the person have chills, fevers, and they come out of it, and they move on with their life, and a few minutes or hours or days later, they will have another chills, fever, so on and so forth. Okay, so what happens, I'm not going to go over details of the signs and symptoms. What happens, some of these guys become, some of them are trophozoites, feeding state, they get into red blood cells and multiply. Some of these guys become microgamete and macrogamete. Are you following? Mosquito comes and take a blood meal. Here, saliva of mosquito transmit after opening up human skin. They transmit the saliva, they transmit the sporozoites in here. But what happens when a mosquito take a blood meal from human, they ingest both macrogamete and microgamete. In the gut of the mosquito, which you do have in 2x2s, I showed it to you in the lab this morning. So in the gut of mosquito, what happens, the female gamete, usually it's a macrogamete, and the male gamete becomes flagellated or pseudopod, right here pseudopod. Then they go together and fertilization occurs called oocyte, which I'm not worried about, but I call it oocyst. Right here is the oocyst. And then in the oocyst, you have sporogeny. In the chicken, just right now you learned in case of Eimeria, I don't want to confuse you guys, the sporogeny occurs in the environment, out in the dirt. Is that right? I talked about Eimeria. Here, sporogeny occurs inside of the mosquito gut. And you know what sporogeny is? We're having a quiz over today. Right? I talked about it. So, comes out of sporogeny are your sporozoites. Sporozoites are ready to be transmitted to human by saliva of mosquito. I hope I'm making some sense. If I'm not making any sense, let me. If I do not make any sense, this weekend I will go to Golden Jail to jump.
You have so much to learn.
Or you guys will have to learn. Oh, does it make sense? Study it, go over it. I'll ask questions. Because it is important. You know, there is more than this, but it is important that you would know the life cycle. Yeah. Yes.
So when the mosquito pees on the bottom of that picture, does it have to ingest a macro and a micro in order for it to be active? So if it only got a macro gamete, then it will.
Nothing will happen. But there, imagine inside of human there are thousands and millions of these guys. Yes, sir. Same question or different? Okay. Here it is, the mosquito gut, and I have them right here. It's the oocyst, oocyst, oocyst. They're all of these big guys are oocysts. Cryptosporidium parvum. As I said, I will come back to it. Here it is. Cryptosporidium live in small intestine and respiratory epithelium. Direct life cycle, does not need a mosquito. Somebody who has diarrhea and they defecate, they go to the bathroom, they don't clean up, and they come and wrap up a taco or burrito for you. Oh boy, horrible. I mean, don't talk like that. Then we starve to death. Maybe you needed to lose a few pounds. Oh, no.
Taco Bell ruined forever.
Okay, but anyhow, that's how, or what happened in this case, what happened in Milwaukee will come up. The oocyst is about 4 to 5 microgametes and macrogametes, oocyst in soil or water, and here, outbreaks in Washington, D.C. and Milwaukee. What happened, you guys? The drinking water of the city of Milwaukee, the city of Washington, D.C. got contaminated with animal feces, cow feces. Somehow, some way, the cow feces, they had Cryptosporidium, they got into the water supply of the town, and the filters of the town is not big enough, well, they're not small enough, I should say, to filter 4 or 5 micrometer. That's very small. They filtered Giardia. Giardia is 10 micrometer. The cyst of Giardia is 10, and usually most cities filter that. But if the filter of the city is, the water of the city is contaminated with Cryptosporidium 4 to 5, that's not the filter they filter. But anyway, that's how Milwaukee, as I said, it is more devastating than Giardia, and a few people died in both outbreaks. In both outbreaks, a few people died. So if a person has Cryptosporidium, definitely they need to be hospitalized. With Giardia, no, you go get some, I don't know, some Pepto-Bismol, you just, I don't know if Pepto-Bismol treated or not, but you know, it will go away. They suffer a few days, 3, 4, 5 days, depends on you, but this one will not go away. It will go away eventually. Last phylum, okay, Phylum Cillophora, mostly free living, some parasitic, some commensalism, and some are colonial. Some sessile, like group Suctorians, they are sessile, and the term sessile, it means they don't move. Okay, and of course, they have to have cilia in order to be placed in this phylum. Larger than most protozoan, if you look at it on the microscope. Plasmodium, no, not Plasmodium, I'm sorry. Paramecium is bigger than the rest of them. Look at it, compare it size-wise. Okay, they'll have multi-nucleated, they have two nuclei. I didn't bring the model. One nucleus is for, they call macronucleus, and the other one micronucleus. Macronucleus is usually for metabolism. Micronucleus is for reproduction. I'll show you some pictures here in a minute. Undulating membrane cilia are fused into sheaths. Cytostome is the mouth, and I'll show you some picture. Oral groove, two contractile vacuole system. I talked about that at the beginning of Protista a little bit. And then passage of water and food in paramecium, oral groove, cytostome, cytopharynx, food vacuoles, indigestible out by cytotract. Reproduction, binary fission, microtubules, micronucleus divide mitotically, it means by mitosis, and you all know what mitosis means from bio 1. And macronucleus elongate and divide amitotically, without mitosis, that would be usually meiosis. Conjugation, when two organisms come together in the lab, remind me on Thursday, I have two slides, you should look at it, of paramecium. One of them is conjugation, and the other one is binary fission. Okay, so I will ask during lab practical, what kind of reproduction is this? It's not in the docu shape. These two, it's a demo slides I have. So one of them is binary fission, like this, and the other one is conjugation, like that. I have it in the lab. And I will, of course, I'll show you some pictures. Autogamy self-fertilization, Plasmodium coli, that's another Cillophora we have in the lab. This one, Plasmodium coli, causes diarrhea. Again, no intermediate host, nothing is needed from person to person, from parakeet to human. A few cases of them have been reported. So anywhere, from any other animal, Giardia. The thing is from human to human? No. Giardia can be transmitted from cow to human. Cryptosporidium from cow, from parakeet, any other animal can be transmitted to human. Okay, it's not from human to human all the time. Here it is. So you have macronucleus, and the label did not, this is micronucleus. Micronucleus, function of it for reproduction. Macronucleus for the arrow. I guess the arrow didn't get there. The macronucleus is metabolism. And then contractile vacuole, I talked about it, two system, you have two of them. And then you have cilia, then what else?